Okay, great. We are being transcribed and summarized. I just want to kind of introduce you. So I'm in Kiel at the moment. I also have a close relationship with the NGS network because we also have CCGA within this institute, and I work very much together with them. You will see some of the data that they created through the years on the presentation today. So this is exactly what it is. We are connecting the layers, and in this case, I'm not going to show you the next multi-omics integration tool. What I hope to take you is into a story of how we have been integrating different omics layers in a more kind of like experimental, mechanistic centric approach. So just a general introduction. So in older times, and this is older times, I mean after 2000s, you could see papers that look like this. This is a Nature paper with some of our colleagues here as well. So you see that the data integration is kind of a simple approach. You normally collect multiple data modalities. So you have an experiment, you phenotype your experiment, and then you kind of associated it with something that was available, whether it was 16S approach or more transcriptome approach, if it could be something like qPCRs, and that's kind of how we like build in the story. However, at the moment, with the advance of next generation sequencing, we are collecting not only data at multiple levels from metabolomics, metagenomics, epigenomics, spatial, single cell, etc., but we also kind of trying to do this for the same kind of simple phenotypic experiment. And this is kind of interesting, but this also kind of creates some problems intrinsic to the analysis of multi-omics. So, for example, you have single cell approaches that capture kind of, if it's not longitudinal, but cross-sectional, like only a part of the system, like a snapshot. And this is interesting to study because you can see, like, for example, how a disease is influenced. However, if you think, for example, in a disease, so in particular, here we are interested in inflammatory diseases, we know that, of course, there's biomarkers that occur in level of the transcriptomics, but we also know that epigenetically already, and this is in the level of methylation, there's still already some mechanistics that are impaired that will lead to a transcriptome difference. And for example, in inflammatory bowel disease, there will be an effect on the microbiome as well. So different layers have different effects. What is cause? What is response? We don't know, and we probably will never know, but we try to kind of tackle this problem on a multifactorial way. So thinking about only the omics, and this is, I think everybody has kind of similar challenges. So of course, the multi-omics themselves, we have heterogeneity. So for example, if you have approach of microRNA or RNA, you can already see that the features that you collect are in very different levels. So you normally have up or more than 20,000 genes in a transcriptome information, while if you only have microRNA, you normally can get around 1,000 microRNAs as features. So there's different scales. There's also kind of like data scarcity. So of course, if we were rich, we could profile everything, but unfortunately, this is not true. So you kind of focus on something that not only you have the ability to analyze, but also something that you have the ability to profile, and it's also within budgets that we all have. So of course, it's not that you can, for example, have all of this methylation, microRNAs, RNAs, protein, and metabolites only in one experiment. Another problem, and this is something that we definitely deal with a lot because we work with human cohorts and also with kind of mouse experimental models, is that we tend to have in the end more features that we have samples. And that could be a problem. It's also like the relevance or insights, and it's just mathematically very difficult to handle once you have very like smaller cohorts, but they have multiple features, and try to identify the features that are truly important for us to further analyze and discuss. And of course, what we have now available, and we have multiple, and I'm not saying anything against, but we have multiple tools that are available, like MOFA, etc. But in a sense, I find it that the results and the insights that we get from these types of analysis are not the easiest to interpret. So you always have this bubble plot with connections between them. Sometimes you have errors, sometimes you have connections, and in a sense, it's very difficult to identify the mechanism behind just bubbles that are different sizes and arrows. So we really try to understand little by little and how to interpret these types of approaches. So what I want to kind of tell you the story is from our own example, and this has been a project that has started in 2019 and has multiple people have participated in this project. Also, I will mention some people here and there throughout my presentation, and has a lot of different omics layers available. But I think it's really important here that these experiments also require multiple people to work on them, not only on the analytics side, which is more my side, but also preparation side, how to prepare the samples themselves. So it takes a village in a sense, and in this case, it takes two groups to try to really do this multi-omics approach to a mechanism. So just a few words of what we are studying. So we are studying protein energy malnutrition. So I sometimes will abbreviate it to PEM. So we know this is the old census. There's been one out yet, but it came out this week and I haven't had a chance to really look through it. But this is from the old census, when we know that there are up to 735 million people that suffer from malnutrition globally. And unfortunately, this normally affects children and women. And we know that 50% of all child deaths below the age of 5 are somehow related to malnutrition. So not directly, most of them, but to have symptoms and kind of like infections that are related to malnutrition in the end. And you kind of have two types of malnutrition. So you know these photos, and I'm sorry I have to show you this, but we have the kwashiorkor. This is a protein malnutrition where you have a kind of this enlarged liver, so this enlarged bellies. And you have marasmus, which is more like a general malnutrition, not only related to protein. And you could see malnutrition not only in people that have scarcity of food and in crisis regions, but you can also see malnutrition, for example, the psychiatric level at anorexia and bulimia. This is such as COPD, etc. So you also have this kind of like with people that have late stage cancer, you also find this kind of incidence of protein energy malnutrition. And there's multiple contributing factors. You could have, like, for example, our patients sometimes have these GI obstructions that lead to malnutrition, appetite loss, inadequate nutrient absorption, etc. So why we studied this mechanism, why we're studying this is that we try to identify mechanisms of malnutrition, and in particular protein malnutrition, that we can potentially treat and alleviate and make it better, not only for people that are in these crisis regions where you have malnutrition, but also people that are in the hospital in late stages. So we do this with a mouse model, and this is just an introduction. So we have our mice and we have our control group where we just give the normal chow to the mice. And then we have a group that we call protein energy malnutrition, which is PEM. And in this case, you have 0.7% of the protein that you normally have in chow, and it's isocaloric. So the mouse has the same amount of calories. So it's only protein that we are targeting here. And not only we have this acute phase of protein energy malnutrition, but we also, after we have this phase here, we also let our mice recover. And then they are recovering with a normal chow diet. So the one thing that we saw we saw multiple things, but this is just to describe you the phenotype. Of course, the mice that are under PEM, there is a decrease in their weight. And then after this acute PEM phase, there is a recovery phase. But what we observe is that even with the normal chow given after the acute time, the mice don't really recover. So there's always kind of a decrease in the body weight compared to the control. Microscopically, so in the intestine, we also know that, for example, the length of the small intestine is decreased, as well as the colon, but only in the acute phase. And microscopically, what we have identified is that Paneth cells, which are kind of the defense cells of our intestinal epithelium, seem to have to be depleted in the acute phase. So this is in the Paneth cells exist in the small intestine. And if you see here, this is a control, this is the acute PEM. They are very much depleted from the intestinal epithelium. And this signature seems to also hold through time. So even when the mice recover from malnutrition, there's still kind of a significant decrease in the number of Paneth cells that we quantify in the small intestine. And this is true for two regions of the small intestine, for duodenum and for ileojejunal. So we could see phenotype macroscopically and microscopically, but then how this really, so transcriptomically, what does this mean? So that's exactly what we did. We took the intestinal epithelium of these mice and we did a transcriptome approach. So in this case, you can see here on the right side, you have the control mice, then you have the acute phase as the second panel, and you have the control and then the recovery with the most on the right. So we are truly interested in this kind of more memory effects. So what we could see is that several genes that are related to the terms such as innate immune response, response to bacterium, apoptotic processes, and inflammatory response are kind of downregulated, as well as these organoid phosphatase metabolic processes, which are mostly related with fatty acid metabolism. So there is, together with the Paneth cells being depleted, and I told you these are kind of like the defense mechanism of the intestinal epithelium, but it seems that there is this general kind of decreased ability to defend against bacteria. The one thing then is like, okay, this is the transcriptomic signatures. Can we also kind of have an idea if there are any epigenomic markers here? So that's exactly what we did. We did kind of a methylation approach on the same intestinal epithelium, and then we try to kind of identify within the start initiate, so the description site initiation, we kind of did kind of a window, and we identified genes that are within this TSS, plus 5 kb minus 5 kb, and we kind of identified genes that were hypomethylated and downregulated, no, hypomethylated and upregulated, and hypermethylated and downregulated. So this we call the DNA methylated linked genes. And we find several genes that do have this pattern. And then once we are analyzing these genes and going back into the literature, we found this gene that was quite interesting for us, which is the GDF15. So going a bit further on this, we have now a phenotype, we have now transcription signatures that seem to also be supported by the epigenetic signatures, but then we only know that this is happening in the intestinal epithelium. So the next step that we did is that we did a spatial approach. And from the spatial transcriptomics, this is very simple. This was still Visium, the version 1. So we had kind of Swiss rolls. These are just rolls of the mice intestine. And you could see we have A1, B1. So we only had like N equals 1 per group, but we could already see kind of interesting things coming from this. So mainly that there were most of the signatures that we find are kind of in the crypts. So in the bottom of the crypts, where also the Paneth cells are. And here we do not only see that there is a decrease in the number of Paneth cells, but we could also see that within these crypts, there is this upregulation of fatty acid metabolism. So going on together, and now here I'm just going to do kind of a little bit of a break. What do we know so far? We know that there's phenotypic effects of protein energy malnutrition. We know that Paneth cells might be behind this, so the cell lineages is altered. We don't know if it's because these Paneth cells are not differentiated or if the Paneth cells are actively dying. This we still don't know. And we know that for some reason, GDF15 axis, there's something to do with it, because of course we saw that GDF15 also is a gene that do not only affect kind of like Paneth cells and their differentiation, but this is something that also can potentially affect the metabolism. So one thing is missing here once we are studying the intestine. And the thing that is missing is, what about the microbiome? So what I didn't told you in the beginning is that, of course, we were from the beginning interested in the microbiome. And what we did is that we not only did experiment with the conventionally raised mice, but we also used germ-free mice. And we kind of did very similar approaches that we have before. So phenotypically, the differences were quite striking, and we were very much puzzled what was happening. So actually, the germ-free mice seem to kind of not have such a decrease in weight as the conventionally raised. So germ-free seems to ameliorate the PAM symptoms when it comes to weight. We also do not see a difference in the number of Paneth cells in the germ-free mice. So is the Paneth cell loss microbiota dependent? That's kind of the question. So how do we analyze this? Of course, there's no point in analyzing microbiome in a germ-free. We have to go back to our conventionally raised, which at the time we had taken a lot of stools from, so we can kind of trying to rebuild what was happening at the time. In summary of a lot of kind of analysis that was done into this, we find kind of like two candidate fatty acids. So for us, we found it nice that it was fatty acids that were our main candidates, because that makes it like matches really well with the spatial insights that we have. So we had this 9-HODY and 12-13D home that seemed to be kind of upregulated or induced by PAM. So you could see here in the conventionally raised, there is this induction. And here you could see that this stays through time and even is increased later on, where you don't see any difference in the germ-free. And of course, the 12-13D home, you could see already an increase here. So in the metabolomic part, we could see that 9-HODY and 12-13D home are quite interesting, but can we see more into this? So here we did metagenomic approaches, and one thing that we saw that are related with the 9-HODY and with the 12-13D home is that these produced by bacterial lipoxygenases and epoxy hydrolases. So we basically can connect now the metagenomic with the metabolomic part, because here we see, especially in the acute phase, there is this increase. What we saw in the microbiome part, and I will not explain here because I don't have the time, is that the microbiome kind of bounces back very well. So there is definitely an effect of the microbiome in the acute phase of PAM, but they seem to recover kind of completely at week 10. So we do not see kind of this more memory effect on the lipoxygenases and the epoxide hydrolases. Furthermore, this is only in mice, but thankfully we did have access to a cohort from the Gordon group, and they also had metagenomics data, and we could see that in human studies, so this was a refeeding study for a group of children from Bangladesh, and we could see here in the human studies that we were not looking in a vacuum, that indeed these lipoxygenases and epoxide hydrolases were also very much upregulated in children suffering from malnutrition. And you could see here what MDCF2 and RUF is. This is kind of adding supplements of the refeeding process, and these are decreasing as the children are being refed. So the cool thing about all of this is that 9-HODY and 1213D home are actually affecting a very particular transcription factor that is called PPAR. There are multiple different PPARs. So you have alpha, beta, delta, and gamma. And this PPAR seems to also be within the kind of the mechanism that will induce GDF15. But this is only a model. So we know that 9-HODY and 1213D ohm is up, PPAR is up, that leads to GDF15 down, up, sorry. So how all of this is kind of making, we need to check, because this, at the moment, it's only correlations. So what we did is that we use what we have at our institute, which is organoids. So the first thing we needed to know is that, can we kind of reproduce the same that is happening in the intestine in organoids, in mini guts that we have in Petri dishes? So this is kind of what we did. So we compare the RNA-seq from the intestinal epithelium with the RNA-seq from the organoid culture, and we see that very well they were correlated. So yes, we can use organoid culture to kind of match and try to identify the long-lasting impact of PEM. And this is exactly what our colleagues did in the lab. So we have several different kind of players here. So what we do is we take the mice, and we take also the PEM mice, but this is like only in control mice, and we grow organoids. And then we have kind of 3 different things. We have recombinant GDF15 that in this case will decrease the amount of GDF15. We have lanifibranor, and lanifibranor is a PPAR agonist, so we will work, on this part without the effect of 9-HODY and 1213D on. And then we also have kind of a 9-HODY molecule that we can use to induce our organoids. So the first thing that we saw is that once you add the recombinant GDF15, there is a decrease at LIS1. And LIS1 is kind of the marker that we have for Paneth cells, because Paneth cells express LIS1. So we could see overall that GDF15 is inhibiting the differentiation of Paneth cells ex vivo. Then we added the lanifibranor, and we saw the same thing. So this PPAR agonist in kind of organoids that are suffering from PEM leads to a decrease in the number of kind of LIS1. So in the number, in the biomarker that we have for Paneth cells, so potentially in the number of Paneth cells. And the same thing when you add the PPAR, so the lanifibranor agonist, we could see that there is an increase of GDF15 in these organoids. So PPAR activation kind of inhibits the differentiation of Paneth cells ex vivo and increases the amount of GDF15 in the medium. So finally, we also tested the 9-HODY, and in this case, we could see kind of very similar to the PPAR agonist. So both are in the same kind of center, but one can work without the other. So in this case, we see once again, 9-HODY inhibits the differentiation of Paneth cells and increases the amount of GDF15. So what else is there to do? And this is things that we are doing at the moment. So there's still a question, where are these oxolipins coming from? Is it from the host and is it from the microbiome? So now these are only preliminary data, but we are pretty sure that the microbiome is the source of 9-HODY. And we did this by spatial metabolomics here in Kiel, together with a group of Manuel Liebecker. And we have now kind of spearheaded this approach. We're still working on it. But yeah, we now have an answer. So you need a microbiome to kind of like have the oxylipins that will induce the PPAR, which itself will induce all the mechanistic changes, so the decrease in the immune system and so forth and so forth. So taken all together, what we have is that protein energy malnutrition leads to phenotypic, macroscopic, and microscopic memory effects. We know that the loss of Paneth cells might be the reason why we have kind of this decrease in the inflammatory responses. So these cells are intrinsic. Their function is to kind of protect the intestinal epithelium against pathogens, and they are also in the crypt. We know now that the microbiome is crucial for kind of having the intensity of the memory effects. So even though the microbiome seems to bounce back, there are kind of like long-term effects, and these are especially driven by two metabolites, the 9-HODY and the 12-13-DHOME. And this is what we think is responsible for this. That leads to the increase in the GDF15, and this increase in the activation of PPAR and GDF15 axis is potentially the possible cause for the kind of decrease in the number of Paneth cells and all the mechanistic effects that come through it. So the fatty acid metabolism effect and the decrease in the kind of inflammatory response of the intestinal epithelium. So yeah, there's still a lot of things, and I wanted to add here a slide, but then meanwhile I forget. There's still a lot of things that we are interested in. We are now trying to find the kind of microbes that are behind this to see if we can quantify them, which this could potentially be used, for example, to study better supplementation for people that are in this crisis regions. We also think that this might be also something that could be used potentially in the hospital once you are within, whether it is like refeeding, and also where you have like chronic illnesses that are leading you to malnutrition. So yes, I keep it short today, and of course, one last thing I want to say to everybody. What I didn't tell you, and it's like kind of a surprise right now, is that, of course, we did not do one profiling. So we didn't do like transcriptome, and then we went methyl, and then we went microbiome. We didn't do that because that's kind of like, that's not how we work as scientists. So we did everything, and we had all the data sets, and we're trying to find things that are in common and to come up with a story. It was the work of kind of years of going back and forth and testing and doing all omics layers and having basically discussions with 5 people. That's, of course, we are a bit biased and we say, oh, sorry, transcriptome is better or microbiome is better, but we need to come to a consensus. So instead of doing a MOFA approach, we did a lot of people approach where we put all the omics together. We try to see kind of mechanisms. One cool thing that I did behind was I ran all our omics layers through a process like MOFA for integration of multi-omics, and they gave me a lot of errors because our cohorts were so small and we had so many features. But in a sense, there was kind of a hint for the PPAR as a feature, as an effect, which it is like crucial in our mechanism, but it did not give me the complete story. So this is also something that kind of a tale of not only looking at MOFA. MOFA can give us like really nice tips, but then we need everybody together to think about the problem and try to identify the mechanism. So yeah, I just wanted to, of course, thank everybody that has been behind this project. And there's a lot of people, not only from here, Kiel in IKMB, but also people that are now in Tokyo Medical University, Harvard University, etc. And also now a shout out to the new people in the project, which is the people from CAU, like Manuel Liebeke, Mathieu Rossin, Mathieu Poyer, Anna, Malin, etc. So thank you very much, and I will take questions now.